I saw like some posts somewhere about that, like how to. You can do that. I think so, but I haven't looked into it. Oh yeah. I am an adult, and like I tried my mom. Yeah, really, seriously, I know what's up. See, that's what I use if I need whatever. She was tracking in high school, and she said, "How do I use this?" Oh, no, you're like, I'm not telling you. When I got people, it's like the method. That's a Lord, yeah, that's so good. I don't use time enough for my kids because Apple has made it nearly impossible to monitor cell phones for minors. Like, yes, with Android, I can get like, they have an Android app. After I just got a bullying profanity notification, have a keyword text message. So, but it's a lot harder on Apple. Right, I don't know what these new ones, yeah. That's what I told my son. I was only 19 when I had this, so it hasn't been that long since I was actively this. So I need to watch that. All right, anyways, so let's get started with this lab final review. We're going to walk through the tests. I'm going to give you as much information as I can. I made my little notes for you guys as well in the notes that I posted from the review on Monday. That was, God, how was it? Just, it feels like it's been weeks, but that was yesterday, I guess, that I did that. Anyways, I posted my notes from that, and I just made my notes from just pictures that I pulled from those pictures available on the module for the lab stuff. There's plenty of pictures and explanations of our tests there. And then I just went through and remembered as much as I could on it. Those notes are, they're missing a couple of little blips of information, like the fact that the SIM assay is a semi-solid butt that we stab. And I didn't put that on there, but I would hope that you guys would know that. I'm just saying, that's a pretty easy one. So little details like that, but all of the big details are on that. And then of course the slides that I actually made, the colorful ones with the pictures, those have all the information that you would need to know there. So use those as your resource for studying. Watch that video where I walk through the tests, see if you can follow along and keep up with me on that. All of our lab Kahoots are up. This is up to, originally when I posted it, I posted the wrong link to it. I was notified that I was posting the wrong link for the Kahoot. Now it is up for this, what we're about to go over. So if you guys want to review that way, you can too. Okay. I feel like Kahoot is very similar to actual tests. It's pretty similar. It's pretty similar to the types of format. Some of the test questions, there's a handful that are going to be a little bit more critical thinking, but it's the same concepts. Yeah. So it's 24 to 26 stations. I say that, that's probably not accurate. I forgot that I share my lab with another class now.
So I have to leave one of the tables open. I can't have anything at one of the benches. So it's going to have some of the stations will have 3 questions. Okay, so it'll be less than 24 stations, but we'll make it like 20 stations, but it will be still 50 questions spread between those stations. Still 1 minute. I'll probably extend it because we might have more 3-question stations. It might be like 1 minute, 20 seconds or something per station, just to give you more time at the 3-question ones. Because it's based on stations anyways. And then it's still Scantron. You'll need your number 2 pencil. I'll have a few to give to loan to you guys if you need it. But I do have your Scantron sheets for you. You get a scratch paper. You know how that goes now. Same rules. All right, so let's start with some basics. Let's get our head in the game. What is most commonly used to inoculate cultures from the choices below? All right, obvious. Thanks. Loop. We got it. So we're going to inoculate with the loop. Another one would be a needle if that were on the list and it weren't. In which quadrant of a streak plate would you expect to find the most single isolated colonies? These would be used to create pure cultures. Remember, one colony comes from one cell. So all the cells in a colony should be exactly identical. That obvious one, again, that's the 4th one where we have it most spread out. We've diluted the cells most. That question will be on the exam. We've seen it before. It's the same figure as well as we got on the last exam. Know what aseptic technique is in general. Be aware of your lab safety stuff. Wear your lab coat, please. You could potentially get a point deduction for not wearing one. I'm not going to have a question. Originally, I thought I'd have a question that was dedicated to having a lab coat on, but now I just can't afford to. There's too much to cover. So if you don't have a lab coat on the day of on the day of the exam, then you will get it like a 2-point deduction. Okay, have your lab coat. You can still participate, but you get 2-point deduction automatically. Even if, huh? Yes. So be sure you have your lab coat. If you have to borrow from me to get the lab coat, you're still getting the deduction. You should have your own at that point. So be aware of that. Types of media. Here we're talking about broths, slants, plates, and butts. You know the difference between those and how to inoculate them appropriately. Let's get into the stains. This should be review. What is the first step in the gram stain procedure? This is talking about the stain portion. Obviously, you would want to heat fix first. Alrighty, so this is going to be crystal violet. Sorry, I'm bringing a brain fart right now. Too much stuff in life. What color do properly stained gram-negative bacteria stain in the gram stain procedure? So our gram-negatives, don't pay attention to the picture. It's just something I put up there. I actually don't know where I got this picture from. Probably was about something at some point. All right, so our negatives, negative color is red. The other option for our positives, purple. And we saw them in the microscope on the midterm, so we have an idea that you should be able to tell the difference between those two pretty clearly. What is the name of this staining procedure pictured? This is a pure culture. So notice that we have like the bubble-like structures, and we have some where we have that tealish, bluish color within the red structures, kind of, where they're not a bubble. So this is the endospore stain. The green structures are the endospores. The red ones are our vegetative cells. Which of the following bacteria would be acid-fast positive? This is a picture of an acid-fast stain. We have a mixture of things here. Those reddish, pinkish guys that are kind of real small, but they're there. Those are our acid-fast positive guys. They'll be that reddish pink fuchsia color. So this is always going to be mycobacterium. This is because of the presence of mycolic acid, which is a wax on the surface of these that are going to interact with that lipid-soluble carbofuscin primary stain. That's our primary stain. So now we have to think of what that counter stain would be later. So if you heat fix your slide and you get going with your staining procedure, when do you use heat again for the acid fast stain? Right. So we don't. On acid fast, we don't use heat. Yep, yep. So that's a little bit of a trick question. So be sure you're reading our questions and really taking that time to be confident in your assessment of the question too, right? So what color is the negative acid fast stain? We talked about mycobacterium being mycolic acid, the wax, the lipid soluble carbofuchsin, and the fuchsia. But what are the negatives? So those guys are blue. That's our counter stain is methylene blue for that one. Where we have that acid alcohol that we'll be doing the rinse with. It's our decolorizer for that one. What's acid fast? So what is the name of the primary stain used for the capsule staining procedure? Now this is an image of capsule stain. We see we have purple background. We have pink little cells. You see we have white around all those cells. Do these guys have capsules? Yeah. If it didn't, we wouldn't see any white. All right, so our primary stain is Congo red for that one. In which staining procedure do we use steam? So there is one where we use heat in the form of steam. Which one is that? All right, so this is the endospore stain. Cool. So know the procedures and the steps involved. Why do we use steam in that one? It helps force the malachite green, which is our primary stain, into the endospores themselves. Vegetative cells won't hold on to it and they'll just rinse clear with water. So remember this procedures, the actual stains used, the order they go in, and why each one is used. And be able to identify them based on just looking at a slide. We're going to move into our differential and selective media next. Which type of media is pictured here? All right, so this is EMB. EMB, we have that dark colored, it's like a dark reddish color media, and then you can have the presentation of lactose use. Being regular use of lactose would be pink growth to purplish growth, but once you get into aggressive lactose use, you have that metallic green sheen that will develop. And only EMB does that. What is the selective agent in EMB? I brought my drink first. Okay, so this is eosin methylene blue. Don't worry, you're not alone because a lot of people are picking biosalts because we know two things that are similar about EMB and the other gram-negative media is that they both detect lactose use. They both have a kind of reddish color to them. The lactose use has that pink color shift for both of them. And they both have like a reddish color of the media. Something else I was going to say about it. Lactose use. Oh, it's gram-negative too. I mean, that's a shared thing. They select for the gram-negatives. So that part can get a little bit fuzzy. But if it makes that green sheen, it's EMB, and EMB selective agent is EMB. Okay, so eosin methylene blue prevents the growth of gram positives. So the reason I didn't mention the name of that other media, guess what's coming? So the other media that's associated with lactose use and pink growth Can't guess this isn't EMB. All right, so this is McConkey agar, this one. So it's also going to select 4 gram-negative bacteria. With McConkey agar, there's a question. I need to keep my mouth shut. Let's let you guys figure it out first, and then we'll talk about it. Is McConkey agar selective or differential? You can choose more than one here. All right, so McConkey agar is both selective and differential. It's not enriched, aside from enriched. So it is differential and selective. Differential because of that lactose color change. Selective because it's going to select for gram-negative enterics. The reason we're targeting the enterics, this guy has the bile salts in it. Okay. That's McConkey agar. Bile salts, gram-negative, enteric gut bacteria, bile gut. In lactose use, this one only goes to pink though. It doesn't go metallic green. All right, next media. This one, I'm sure you guys you'll recognize it. Hopefully. Hopefully. All right, so this is mannitol salts agar. We saw that there was like that coral reddish orange color of the media itself, and then we had that yellow quadrant where that growth was. So we can look at that and should probably be able to guess that the pH indicator for that media is phenol red. We get pretty familiar with it for this semester. So I'm going to go through our pH indicators. We only have 3. I know it seems overwhelming with all the different types of media, but there's only 3 pH indicators, okay? So our first one here, we're introducing it with our mannitol salts. This is phenol red. Neutral, it's at that coral orange color. When we have acid production, it's that bright yellow. When it's alkaline, it gets to a vivid pink color. We'll see it over and over again. We also have salt, literal salt, sodium chloride, that is present in mannitol salts agar. That is our selective agent. It favors gram-positive bacteria because they have that thick cell wall. All right, what is the pH indicator used? I always forget this. So what is that pH indicator? We see it turning yellow because we are fermenting the mannitol, which is an alcohol sugar in the media. Staph aureus can do that. Now, if we talk about other staphylococcus bacteria, remember Staph aureus isn't necessarily the only one. There's other ones, and any of those staphylococcus media those staphylococcus organisms, whether it's aureus or epidermidis or other species, they can all grow on staphylococcus media that is not differential media. This has the same salt content as that, but it also has our pH indicator that helps us get a little bit more differentiation. All right, be sure you know which of these media are selective or differential or neither or both or enriched, maybe like a blood agar is. Know the selective agents for those that are selective and what makes them selective. Know the color changes and what they mean. The pink growth for the lactose use or the aggressive being the metallic green and EMB only. As well as our pH change colors and what they stand for. Any pH indicators that are in that media, really only going to be mannitol, and it's going to have that phenol red. We didn't talk about staphylococcus really that much other than what I just brought up. It is selective. It is not differential. Whereas blood agar is enriched and it is differential, but not selective. Differential because it lets you know the different types of hemolysis. If you grow on blood agar, you should have especially on your little sheets that I gave your comments on at 1:30 in the morning. You should have a type of hemolysis associated with it. If it didn't hemolyze, you're like, well, it just grew, it didn't hemolyze. That's gamma hemolysis, okay? So be sure it should say alpha, beta, or gamma. Alpha, you'll have greening of the media, which nobody had. Or beta, you'll have true clearing of that media from the total breakdown of the red blood cells. Beta, hemolysis. I'm trying to think of that. Remind me of one other thing that was on those sheets that almost everybody got wrong. The capsule. a lot of people were putting results for our gram-positive organism in the capsule. We did not ever test that. So there shouldn't be a result should say not tested, or it should say NA. We shouldn't have had blanks on there because we either just knew that we didn't test it, or we were negative, because if you're testing for acid fast, your gram negative is always negative. So we just kind of knew that. Don't forget that we did actually look at colony morphology, and we learned about it at first in unit exercise 2. We learned about the terms. We looked at our mixture, but you can still get those terms from the exercise 2 and apply it to your nutrient auger from exercise 11. We should have 3 terms per organism for that. At least just 3 terms. Man, if I was being stringent about this, it should be you have an example of at least 1 from each category. There were 6 categories, but 3 is all I'm asking. Maybe something simple like just the color of what it looked like. And then the other one was solid media. It can be, you can get the information from any solid media. The best one is going to be your nutrient auger one. Okay, that's that means, yeah. And then the other one was the motility, which I had a student try to have an argument with me about in my other classes. She just don't want to try to do. So she was like, well, we did the motility on our positive. So I just put that result in the motility at the top part too. But that top part specifically says wet mount motility. So we did not test the wet mount motility for our gram positive. We only did the SIM motility for our gram positive. So it's not the same test. So this is part of just paying attention to what the form is asking of you. And again, that's kind of why I made that little clip at the end of the announcement about get your edits in by Wednesday at midnight. I'm letting you guys do edits, get your points back as much as possible. If you put something and I said made suggestions, put something, get your points back. I'll change them. If you want, yes.
I just have a quick question about the italicize.
Yeah, if you can't italicize it, don't worry about it. Okay. Yeah, because I know some people's computers aren't letting them.
Any formatting, any font change.
Yeah,
nothing.
Most people can't. I have a few people that figured it out, but don't feel pressured. Just as long as you capitalize and lowercase, right? First word has to be capitalized and the second word has to be lowercase. If you're doing it by hand, I don't know if you can do it with the, you know what I'm saying. You can't italicize, I don't think you can. But if you're doing it by hand, some people literally turned their forms in that were handwritten, even though it says not to do that. I would prefer that you not. Because that is really hard for me to read, and I actually make comments like that on people who turn them. If you had to, if you did it, I may do, but like some of them I just literally couldn't read it. So you get points off. You were not supposed to. Yes, you're not.
Okay, so we didn't do capsule on the gram positive.
Correct. We only did the negative, and we did not do the wet mount motility. Yes. Any other questions while I'm here thinking about that? So I think those were the biggies. The colony morphology, some people just put growth. So I want actual colony morphology descriptors. There are 3 for each organism. We had a lot of people that left for whatever reason. I know it's a lot on the form, so I think some places just got left blank because they just didn't quite see it while we were filling it out, and it happens. But if it got left blank, the unfortunate part of that is I need to count off for it because we do have people that turned in very partially completed forms that I think we're hoping to get credit back from. We're like leaning on getting credit back for making edits later. And I just can't have that for people that did all the effort to fill theirs out. Yes. That is the only one that can be blank. Yes.
Yeah, that's what I did fill in because I think we were told what it would have been.
Right. If you filled it in and it should have been blank, you can still go back and check that and get, that's fine. I'm probably not going to be super picky about that one if I'm being honest. And for blanks that you may have left for ones that we didn't actually do the testing, I'm probably not going to be super picky about that either. I mean, who cares, right? But I'm making the comments. So now you know, and if I did my part, now you guys get to do yours. The biggest also other one was that the descriptions on that last page, a lot of people did great with them. And then I have a lot of people that didn't. The descriptions need to be as much information as we ever learned in class about that test, all of it, okay? And described in a way that shows me you understand it. So saying that this is a test that's done to show me about metabolic differences in organisms and leaving it at that, not acceptable for that last page. So if you go back to my announcement, it literally says how to fill out that last question. It has that in, it's like unknown key, and how to do the last question. I have my description of urea, which is an easy one. You cannot use urea because I used it and because we didn't actually do it in class. It says it in the announcement, so don't. So if you need to change it and you used urea, please go change it to something else. Use a different thing and describe it in at least as much detail as I do for urea. That is a baseline, not the above and beyond. Okay, yes.
Is there a way that we can go back to the, like, on Canvas to look at, like, let's say we need, like, the reagents for the nitrate reduction test? To see which specific reagent.
You don't need to know if it's not on our sheets, ever. So those descriptions that you guys are doing on that last page, a lot of people I can tell went to nice old chat GPT and put in whatever it told it to.
I went to Top Hat.
Yeah, wherever you went.
Our-
Top Hat has a lot more info in it as well, but on our sheets, those fronts of those sheets, that's as much info as you need to know. If you put in extra, you're not going to get points off for that. Okay, it's just I'm telling you that it's extra work. But a lot of people did that. Gave me all these descriptors of the names of the A and B that we use in nitrate, or like the name of the actual reagent for the oxidase, and went into all these chemical reactions and explanations. Y'all cannot possibly tell me that you understood what you were writing out on that. You did that. So you're just copy and pasting. I'm not going to give points off because you did cover your stuff.
Whenever I was typing it up.
Yeah.
I just did the two that I remembered.
It's fine. So we know we need to get at least as much detail as we had to know. If you're covering citrate, you should cover all the info that you could possibly get tested over on our test.
Okay.
That makes sense? So if you have to know it for the test, it should be in your description. So some of them are just missing like a detail or two. But I pretty much, because I gave as much personal feedback as I could within reason. But again, I have 78 of those. And so as much, commentary as you might have gotten on yours, I had 78 of them. So I'll try to be reasonable. You submitted it by Sunday at midnight, but I wanted to give you guys time to edit it, and I don't want to have to keep grading them over Thanksgiving week. So you get till some leeway. I'm being nice enough to give you some changes to get those points back. So Wednesday at midnight, please. Yeah. If you have any questions about what exactly I am looking for, feel free to email me and I'll get back to you.
And Thanksgiving next week.
Nothing for my class. Yes, do not show up to my class next week.
Okay, that's what I was.
Yeah.
Double.
Because I don't want to be here. You don't want to be here. From my class, take a break. Sorry, you can't for other classes. I don't know what to say for you. Sorry, my tears will be for you while I'm on in my pajamas at home. All right. That's the differential selective media. So we still got some more stuff to truck through. Now let's get into our tubes and that stuff. Which test is this one that's going to be in this picture? All right, so this one is the citrate test. We know this is citrate because the citrate was our slant that was green that turns blue, right? So that's kind of part of what the next question is going to relate to. Let's see. Which of those tubes was positive? So our positive one was the one with the blue in it. So that's the one that was on the right. There's B, tube B. I don't remember if the next question was about the pH indicator or not. Yes, it is. Okay. So what's happening to the pH in tube B? Okay. So tube B, our pH is going up. Remember what a base is? Alkaline means that you have an increase in your pH. So remember your pH scale because that absolutely is going to come up more than once on the exam. I will have a tube. I will sit it in front of you and just say, what happened to pH? Tell me what happened. More than one question on the test for that. That was Simmons citrate. That's the name of it. How do we inoculate that one? That one's just a slant. So we're just going to do a surface streak for that one. How do we read it? We're looking for color change. It is green to begin with. That's our neutral for the pH indicator, which is bromothymol blue. If it turns blue, it is alkaline. The alkaline product comes from breaking down citrates into a carbonate as well as ammonia compound, leading to that alkaline shift and therefore that blue color. Okay, so we had phenol red. Phenol red is that coral orange color in neutral. It turns yellow for acid and pink for alkaline. Easy. Bromothymol blue is green when it's neutral. I don't care about its acid color. Its alkaline color is blue. In fact, in our next pH indicator, our last pH indicator, it's going to have a similar effect with the blue being alkaline. If you were looking at phenol red's colors and you had to choose the bluest of those three colors, yellow, orange, and pink, which one's the bluest? Pink, right? It leans more blue than yellow and orange. Blue is base. Okay. So if it's any blue in your pH indicator or shifting towards that blue spectrum, alkaline. Base is alkaline. Choices are alkaline and acid. Obviously a base is an acid. Okay. Your pH scale is up there to remind you, 0 towards acid, 7 is neutral, 14 to alkaline. And you better believe that's going to be on your lecture final. pH stuff's coming back.
Well, we need to know about, I get that. I always get confused about, like, it releases hydrogen positive.
Not for the lab stuff,
Okay.
but potentially for the final, that could be on there. The lecture final, that's what I'm saying, yes. Not for the lab final. All right, which test is this one that's pictured? Interesting, so we have a tube with another little tube inside of it. And it's an orangey coral color. All right, so that is phenol red carbohydrate fermentation. We only have two tests that have the upside down tube in them. One of them is carbohydrate fermentation, which on the exam, there will be a question about carbohydrate fermentation, at least one. And I will not call it phenol red carbohydrate fermentation. I will call it carbohydrate fermentation because you're going to, of course, have to know that it's phenol red, right? I think the color gives it away. But, you know, because what are we looking at with fermentation? Acid and things like that, pH change. The other one that does not have that orange color is going to be like a more watered down regular muted color. That's the nitrate reduction test. All right, what is the name of that upside down tube? Easy one. Okay. Durham tube, you guys got that. It's not a surprise. That'll be on the test too. Yeah. All right. This one could be more than one. What are the two products, there's only two, that carbohydrate fermentation test detects for fermentation? Looking at fermentation, what can you make? What is it detecting in that? All right, so carbohydrate fermentation test, we have phenol red in there to detect whether or not you're making acid from fermentation. If you ferment, you make acid for sure. If you ferment, you can also make gas. But if you make gas, you will have also made acid. Okay. What other thing could you see in that test is pink. If you see pink, then it is alkaline, and that's from breaking down the peptone, which is our nitrogen source. Makes ammonia. Ammonia is notoriously alkaline. Covering all our bases. All right, which test is this one? This is one test. I want you to take note of what's going on at the top of two and three. We'll talk about that in a little bit. It'll come back up. It's not the last time we're going to see it. All right, so this test is the SIM test. Okay, so SIM is a semi-solid butt. How do we inoculate it? Stab it with a needle. All right, what does the I stand for in SIM? Brittany, that's what we're here for. Yeah. Again, you'll have choices.
Looking at all the words.
Right, then it's obvious, yes. That's why we don't have the short answer questions. This is indole, right? So there is another test that you guys tried to think this was at first, right? The TSIA test. So this came at the same time. We learned about it at the same time. This one is the butt one, and the other one's that orange slant, right? So the TSIA. What does I stand for in TSIA? Iron. So what's the iron doing in that test? Uh-oh.
It's the sulfur thing. Like
Yep.
if it gets dark, it says
Yep.
that there's sulfur.
Exactly right. So the iron in the TSIA equates to the S sulfur in the SIM. It's the same thing, but the iron in both of these binds with the H2S to make the black precipitate. It's just we don't have the iron in the name for this one. I for this one is indole, right? No indole in TSIA. That one's just iron binds with the H2S, makes the black precipitate. Yeah. All right, so how do we detect indole with this one? So this shows this part of the test in the second two tubes. The comparison between positive and negative. In the third tube, we had that golden layer. That was the negative. Adding for indole, we added Kovacs reagent. Kovacs reagent turns that bright red color if you have indole. Indole is a product of tryptophan catabolism. So breaking down tryptophan. That's going to be on the test. Okay. Yep. Remember? Some guy's name, yeah. Right. So how do you detect H2S in SIM? It's the same way that you detect it in TSIA. So when I was a little kid, my parents used to call me Davy. I liked the Monkeys, and the lead singer was Davy Jones. So they called me that, and also because my last name was Davis, that's my maiden name. And like I'm always considered myself a Davis. And both times I got married, I took their last name, and I just hate it. That's not me. You guys know me as Yost, but that's I'm like that's not me. I'm Davis. Right. That just made me think of that. Black precipitate. That's our sulfur. Right. How is motility detected in a SIM test? I think of sulfur as being like an evil villain smell, and then the black is like an evil villain color. They often wear black to costumes and stuff, so that's how I remember that. Motility. So if we look at the first two tubes in that picture, did you guys see a clear stab line in that? They're pretty cloudy. So if it's cloudy like that, you can't see a very clear defined stab line, or you clearly see the growth is clearly deviating from the stab line. That is motility. Precipitate. There you go. Whatever helps. So SIM, we've talked about inoculating it with the needle, stab, it's a butt. Interpreting the results and changes, we've kind of gone over all of those in this. And Kovacs is the reagent for this one. Which test is this one in the picture? All right, so that one is litmus milk. That one's pretty easy one to get. It looks like purple milk. All right, in litmus milk, what color is associated with an increase in pH? So increased pH, base. What's this rule? It applies to all three of them. litmus is our pH indicator for litmus milk. It's literally just litmus. When litmus is neutral, it's lavender. Whenever it's acid, it's pink. Whenever it is alkaline, it's blue. So blue is truly our rule here. So the bluer it is, the more alkaline it is, okay? So we're gonna hold on to that. Litmus milk, though, it gives us more than just pH and effects of just the pH. So that one gets a little bit more complicated. I'm gonna draw that one out on the board as well. Use that board, but I don't really want to turn the screen off. So you guys can see this. Everybody can see this. Let me try it with this. Bear with me because I don't have like a screen to write on. It's gonna be a little janky. All right, so with litmus milk, we'll start here in the middle. This will be our middle, okay? Way, way out here, we're heading more towards our base. This will be like an upper end of our pH scale, base. And then this will be towards, I want to move this, we're going to move this acid out here. Okay. We have a couple spots along the way on each side of this. So this one is going to be our neutral. So this is our start point. This is neutral. Definitely can read my handwriting. It's really good. That's why I hate using the mouse. The pen is so much easier. I don't have a pad here for me to use. Okay, so neutral. If we go towards the basic one, right? What we're doing here is breaking down casein. So this is casein, C-A-S-E-I-N. It's a protein in milk. All right. So breaking down casein, you can put catabolism if you want. That's what breakdown is when we break something down. Or metabolism, that's summarizing that we're using it, okay? So casein catabolism, our first stop is being alkaline. Put alk, save me the trouble, please. Our neutral was lavender, I'll put an L for that. Our alkaline is blue, we just did that question, right? If you keep on breaking down those proteins, so we have actual complete breakdown. Complete breakdown of those proteins. Now we're going to make this turn into digestion. Okay, come on, digestion, okay? And this one is transparent. I'll put a T for transparent, okay? You can start to see through it. Now we'll notice when we look at digestion, typically it'll have that like plasma color to it or like regular media color. It's like that off clear color, yellowish clear. And at the top, it'll have like a purplish layer. The litmus is still there, okay? It hasn't changed the litmus. Litmus is still hanging out, but we just got clearing up with the proteins. So that's happening as we go towards breaking down casein more and more and more. On the other side of things, now we're not talking about breaking down the protein, we're talking about using lactose. We're just going to put lactose here. This is the sugar that's just inherently in milk, right? So if we use lactose or catabolize it, this could be fermentation, for example, breaking it down. Our first stop here is acid. So if we look at our little chart, what color do you get when litmus is acidic? Pink. We'll put pink here for P for pink here. So when you were just starting to ferment it, if we're really aggressive with this, kind of like we got the metallic green with EMB for aggressive lactose use, if you're really aggressive, so we're going to go aggro here. Then we're going to have reduction. Okay, so we are actually reducing litmus. So litmus gained an electron and was reduced. If we're gaining an electron, we're changing the chemical composition of litmus. Litmus was all these colors. But now we're changing it. It's not litmusing anymore. So we don't see it anymore. So now we see the white of the milk. So this one is white. Okay. You look at the white from breaking down the lactose and lead to reduction of the litmus itself, or you look at the pink from breaking down the lactose and just having acid production. Either one of those can lead to coagulation. This is like curdling or solidifying. And you can have coagulation with gas. Not always, but you can have gas. If we had gas, we'd break up that curdle and see the pockets of gas as well as the breaking up of the actual media. Okay. But those both can happen with the pink and the white. You don't have to have coagulation when it's white, but you can.
So is digestion and fermentation like opposite of each other?
Yes. All right, we good on that? Is that helping out a little bit? I feel like this is better than the tables.
Way better.
Okay. My handwriting is terrible, but yes. Okay, let me get rid of this then. I wish it would just like have a button I could just clear all drawings. There we go. Okay. Next question. We're getting there. I promise we're almost there. Not as much explanation in the coming ones. So in litmus milk, the white, what does it mean? We had that in the drawing I just made. Let's see if there's paying attention. That drawing is also in the summary sheet that I made from Monday. Those drawings, it's there too. All right, white is reduction. So actually gaining the electron from the litmus, litmus doesn't litmus anymore. We kind of went through all the colors and what's happening with digestion is breaking down all of those proteins in the milk. We talked about the gas. What's the sugar in milk? Lactose. There might be a question about what other tests also deal with lactose, so it's nice to know that stuff, as well as phenol red, like what of the tests have phenol red. So this is not the original color of the media. We added something to it to turn it this color, and then these were results we saw after adding the thing. My favorite question for this particular media on the exam is what do we add to it? Like what reagent? So this is starch hydrolysis. Okay. Originally have regular old color media. We add something to it to get that dark color. So in this test, which one was positive for starch hydrolysis? The one on the left or the one on the right? Or were they both? So this one is actually the one on the left. We've got that halo from the breaking down of the starch, and the thing we add to it didn't react there because there was no starch there. The other one, we could just see where the growth was. So again, the question you'll probably see is what do we add to detect starch hydrolysis? All right, so this one is iodine. You guys did great with that. All right, which test is pictured here? You see there's a little tube inside of this tube, right? So I told you there's two tests that have Durham tubes in them. This is the other one. This one is the one that is not orange originally. It's this off color. So this is nitrate reduction. It's just those two that have Durham tubes. What do reagents A and B detect? It's your favorite. You know what A and B detect, and then you know what zinc detects. If you can remember one, it should help you with the other, because we're really only testing for two things, two products. All right, so A and B are detecting nitrites because guess what this one's going to ask? Excellent. So if you can remember that, you write that down on your scratch sheet and you know you're looking for nitrate or nitrite. You know zinc is nitrate because it comes late, they both do. And so the early one will have to be nitrite. That's the A and B.
What is the actual difference between nitrate and nitrite? It's some kind of chemical, less electrons.
Oxygen, so there's another oxygen on it.
Extra.
Mhm.
I know that-
So nitrite is,
somehow that helps me remember.
yeah. So if you had something NO2, that would be nitrite. If you have something NO3, it's nitrate. Chemistry, if you're interested. So we know we have our little mini test of the nitrite and the nitrate, the A and B versus the zinc. A and B, red, positive for nitrite, therefore you definitely reduce nitrate. Zinc, then you are testing for nitrate. In that mini test, you are positive for nitrate, which means in the overall test, you are negative for nitrate reduction because you still have nitrate. Yes. So nitrate reduction implies you are not going to have nitrate if you have reduced it. Just like with litmus, we reduce litmus, not litmusing anymore. I'm not going to ask you about the gas on the test. What test is pictured here? Definitely one test, believe it or not. C is control for the original two. Alrighty, this is TSIA, triple sugar iron assay. Which sugars are tested for? Sucrose, lactose, and glucose. Excellent. You guys are learning stuff against your will, but. All right, which of those tubes shows evidence of H2S or dihydrogen sulfide production, AKA sulfur reduction? A lot of reduction going on in these tests. Sulfur-based stuff in these tests, what are we looking for? All right, so for sulfur reduction, regardless if you're SIM or TSIA, where you're looking for black precipitate, those are tubes 3 and 5. Similar kind of question, we're looking for gas now, but we don't want H2S. So which tubes had gas that don't have H2S? Okay, easier one. Alrighty, so that's gonna be tubes 4 and 4A. They had air pockets underneath the media that literally pushed the media up. So that's actually happening from gas production from fermenting those sugars. This one, you do need to know the colors for the phenol red for the acid, neutral, and alkaline. We're talking about fermentation of sugars again. If it's happening in the slant, it's the lactose and sucrose. If it's in the butt, it's the glucose. You also need to know about the black precipitate for H2S, and then gas production. You'll see cracks or bubbles, usually in the bottom of the tube. Yes, that's everything for it, yeah. Yes, the slant part is the sucrose and lactose. The butt part, we're assessing glucose. How do we inoculate this one? Slant and stab. Yes. Or which test is this one? All right, so this is one that we didn't have in our actual labs. This is the urease, but you will get tested on this. I have one question, not a station, but just one question that's going to ask you about stuff you're supposed to know about urease. It will not have any pictures. So it's going to be a theory one. So it's going to be in one of those questions like, What other test also has phenol red in it? And then I'll have like phenylalanine deaminase. Does it have that? You should know that it doesn't. It doesn't have phenol red. Phenylalanine deaminase doesn't. We should know that citrate doesn't, or that staphylococcus auger doesn't. And then if another choice was urease broth, it does have phenol red. It has phenol red. We break down urea into carbon dioxide and ammonia. Again, ammonia puts you all the way down here in the alkaline. So pink. Pink is positive like we saw there. Vivid pink is positive. Original is the orange. That's it pretty much for that one. All right, which test is this one? Everybody gets it split on this one. I bet you guys will too. See if you don't. If I warned this, you haven't answered yet. It usually trips people up. Might make one. Most people are going to pick the right answer. So this is catalase. Yes. So catalase is the enzyme that breaks down hydrogen peroxide. I know you guys know this was the hydrogen peroxide test, okay? But catalase is the enzyme that breaks down hydrogen peroxide into water and oxygen. Oxygen is where we get the bubbles from. Oxidase refers to cytochrome C oxidase. It's a protein in the electron transport chain. It gets that last electron, gives it to oxygen, then we get that H2O as the byproduct. All right, so which one is this one? That one's the oxidase. Good job. So if you can keep those two separated, that one's a confusing one for a lot of people because of the name. Don't get tripped up. All right, we also talk about malonate. If I were to give you basically a liquid version of the citrate test, we're not testing for citrate being used as a sole carbon source. We're testing for malonate being used as a sole carbon source there. Okay, same color changes though, it's in a broth form. Phenylalanine deaminase tests for deamination or removal of an NH2 group from phenylalanine, an amino acid. If you do that, when we add ferric chloride drops, that yellow drop stuff, if it turns the slant a green color, then it is positive for phenylalanine deaminase. Otherwise, it'll stay that yellow color. MRVP is the methyl red Voges Proscour media. There's two tests, the methyl red, the Voges Proscour test. Methyl red, we add methyl red. If it turns red, you're positive. If it turns orange, you're not. That tests for heterolactic fermentation. VP, Voges Proscour, you add 1 naphthol and potassium hydroxide. That was the 30-minute one. If you got a red layer at the top, you were positive. If you didn't, if it stayed that golden brown color, then it was negative. That tests for mixed acid fermentation. Gelatin, we're looking for the enzyme gelatinase that breaks down gelatin. Gelatin was what made that one semi-solid. So if we put it in the fridge and it is still liquid, even at that cold temperature, then you broke down gelatin with gelatinase and you are positive. Otherwise, it'll stay solid and you are negative. Our bacteria basics, get close, getting close. How would you classify this organism's characteristic arrangement? And it has an arrow pointing to the better example of that. The arrangement, remember? Yes. Mm-hmm. All right, so this is the strepto arrangement, so strip or chain. All right, now what is the name and shape of these bacterial cells in this picture? Say the name of the shape. What shape is it? Now you guys know on the test, you're only going to ever see Bacillus or Coccus, and you're only ever going to see Staphylo or Strepto. Those are the only ones you need to know. But you guys know this. Bacillus is rods. We're doing pretty good. So our shapes, cocci or bacilli, our arrangement, Staphylo or Strepto. Don't forget that those are not interchangeable like categories. And also don't forget about simple stains. They give us contrast. If you dye something, no matter what kind of cell it is, eukaryote or bacteria, whatever, if you stain it with methylene blue, even if it's a gram positive, it's gonna be blue if all you're using is methylene blue. Okay. That was kind of tripped people up last time too. All right, microscopes. If you look through the 100x objective and the 10x eyepiece, what's the magnification total? I don't remember if this was on the test or not. I still have 5 questions I need to write for the test, and I have to put it together and everything tonight. So I'm on a late day today. Okay, so this is 1000x. All right, which lens is used with oil immersion? Okay, that's the 100x. That's the ones we use. It was also in the picture, by the way. Know your parts of the microscope. Be aware of lab safety. We went through the stains. We went through the tubes and the plates. Any of the media that we talked about. And then be able to, if I give you a chart, like that was your first page of your unknown report, everything filled out, I'll have one that I would provide you that's already filled out. And it's already filled out. I'll give it to you. I'll give you a dichotomous key. You don't have to memorize it or anything. I'll give those to you and then ask you, what is the gram-positive organism? Or if I were to give you a list of things, it's gram-negative, urease negative, phenylalanine deaminase positive, it made acid but not gas for glucose and whatever. And you get to use the chart, and I'll give you a list of things. Yeah, but it will potentially have extra information. Now, so does the little other chart, right? But sometimes the list of things, people think they need to use all of it. So follow what the dichotomous key tells you on your little route. Always start from the far left and work your way right. And you should be fine. So that shouldn't be a hard one. That's meant to be easy. So there'll be 2 questions like that on there. That's that. So good luck with your studying. Here's our little winner podium. I was notified by one of my students that another student had showed her, hey, look, I found a video or something of Professor Yost, somebody going over Professor Yost's micro exam. Somebody had taken it upon themselves that when I open up the exam for you guys to review for study purposes, that was appropriate thing to post online in a video and go through the questions and answers. If I found out who that person was that actually posted that, you better believe I'm going to take you to the president of the university and get you expelled. That is not acceptable. If I give you the answers to my test and the questions to my test, it is within university policy. You are not allowed to disseminate that. Okay? If you record my lectures without my permission, that's also not allowed. Right? Anything like that. It falls in that category. If I found out who that was. Problem is that my questions, not everybody gets the same test. I use banks. So you might not even have those questions on your test. Second of all, I never open up my final, so good luck with that. And I also never use the same questions on my final that I've used on other tests. I might reword them or rephrase them or something. So don't rely on exact memorizations of that. That doesn't mean I'm hunting out the person that did that, but you might warn people if you have anybody that you know like that. I feel like if we're adults, then we know that.
I would warn anybody, like.
I mean, I'm just saying, if you happen to know that somebody was planning on disseminating my tests, you might want to let them know before I find out. Because I will expel you from this university. I don't know. I haven't seen the video. I was told by the person who found out about it that it doesn't look like it's my actual test. They just thought it was like my test. But even if you did think that was my test, you guys, what is wrong with you? Why would you risk that? You know what I mean? So you could get literally like, if you get expelled from the university, pretty much all the credits that you earned are. Exactly. Nobody's going to ever take you to another university. You'll never be a nurse or anything ever again.
It's
So.
just one test and one final.
How is that worth it? But like I said, I'm not really pushing too much on that. Just if you know people that are involved in that, just give a heads up. Don't do it. Academic honesty, right?
I mean, why would you do it in the first place?
And when you can, I honestly feel like if you learned the material and took the time to do that, that will actually save you time and effort on the test.